Looking at the structure of the mitochondria again, of course the cristae I just talked about, this inside space is called the matrix. And that's where one of our metabolic pathways is going to occur, which is called the citric acid cycle. So I'm just going to draw a big circle here and write Ca, citric acid cycle. So all the enzymes driving this cycle are occurring in the matrix. And then the proteins that are embedded on this cristae are a vital part of the reactions that are about to occur. So we're right here at pyruvate, a 3-carbon compound. We need to get down here into the matrix. So there is a step called the transition reaction that bridges what happens here during glycolysis to the Kreb cycle. The citric acid cycle is also called the Kreb cycle. It's called the Kreb cycle first, and then they've changed the names over the last few years. So this transition reaction bridges glycolysis to the citric acid cycle. And the reason why we have to talk about the transition reaction is because when this pyruvic acid gets transferred into here, a carbon dioxide is given off, and we're seeing this first product being made here. So as this is being oxidized, we are producing carbon dioxide. So it's going to build up in the cell, diffuse out, and then how do you get rid of it?
Breathe it out.
Yeah, you exhale it. I haven't even shown how we use this yet. We haven't even seen the oxygen consumption, the reason why we inhale. But right now I'm showing you right here we're producing some of this carbon dioxide. So during this transition reaction, you're losing carbon dioxide. So a 3-carbon compound loses a carbon. A 2-carbon compound called an acetyl group will react with what's called coenzyme A, making acetyl coenzyme A. That will react with a 4-carbon compound, making a 6-carbon compound called citric acid. That's why it's called the citric acid cycle, again, happening in the matrix of the mitochondria. The citric acid cycle, and it's a cycle, so it will go keep turning. There's all kinds of reactions, and if you look at the picture in your textbook, you'll see that there are many reactions occurring, and I'm just going to hit the highlights here. Oh, during this transition reaction, we also had an NADH made. So we got 2 of them because 2 of these, remember there's 2 pyruvates. So 2 carbon dioxides, 2 pyruvates went in, so 2 of these NADHs. So during the transition reaction, 2 NADHs, we had to keep track of those because those are going to be worth some ATP. So now during this, we're going to lose some carbon dioxide in a couple spots, but we're also going to make some NADH. Let me clean that up. So we're also going to make some NADH in 3 spots. You'll see if you look in a textbook and look at this cycle, you'll see there's 3 spots where we make some NADH. The compounds in here get oxidized, meaning they lose hydrogen ions and electrons, and the NADH, NAD gets reduced to the NADH. So we got 1, 2, 3 for the first turn of the cycle, then 4, 5, 6 for the second turn. So during the citric acid cycle, we have 6 NADHs made. Now also what will happen down here in one of these spots is we'll have ADP gets phosphorylated and ATP is made. It's going to happen once with the first pyruvate that goes through the transition into the citric acid, and then again. So there's going to be 2 ATP made. Have you seen any hydrogen ion reservoir with the channel? Have you seen any oxygen being used? No. So what kind of ATP production was this?
Substrate.
Yep, so again, we've just now seen some more substrate level phosphorylation. So during the citric acid cycle, we've got 2 ATPs made by substrate level phosphorylation. And we had 2 net substrate level up there in glycolysis. There's one more thing that happens here that's going to be important, and that is an FAD gets reduced down to FADH2. There we go. And this molecule will be responsible for the production of some ATP. So because this is only going to happen once here, it's going to happen each turn. We're going to have 2 FADH2s. So we've only seen 4 ATP made, and I said I'm going to show you 36 to 38. I'm going to show you the 38 first, and then I'll talk about the 36. What haven't we talked about yet? Oxygen. We haven't talked about oxygen or really what happens on the electron transport chain. So I'm going to clean this up again, and now we're going to talk about oxygen. As long as there's oxygen present, it, next to fluorine, is the most electronegative element that there is. And what's it mean to be electronegative? Remember way back from your chemistry chapter? The tendency to drag or hang on to electrons. So with oxygen present, oxygen's going to be drawing electrons through this electron transport system. And it just, you inhale it, it diffuses into your plasma, diffuses across your tissue, diffuses into the cell, diffuses down there. This is the only reason we breathe oxygen. There's no other function for oxygen in the body except for right here. And it's drawing electrons through this electron transport system. And what's it drawing it through? The electrons are being, and hydrogen ions from the NADH and the FADH are being taken off and then handed down these cytochrome proteins here. So electrons are being drawn through, hydrogen ions are being pumped across that cristae membrane into this inner membrane space, the outer membrane, the inner membrane. So this is the inner membrane space, building up hydrogen ion reservoir here, and in here you hardly have a comparison. So just through the law of diffusion, which way are the hydrogen ions going to flow? Into the matrix. So you've got one of those channel proteins with an enzyme at the end, ATP synthetase, helps catalyzing the reaction. So you got hydrogen ions rushing through, and that's going to take an ADP and add a phosphate group to it, making ATP. For every NADH that comes through, it comes through up here and gets handed down, the hydrogen ions and electrons. So for every NADH, you get 3 ATP made. For every FADH2, that comes in right here, and you get 2 ATP made. So if we go over here, during glycolysis, we have 2 NADHs. Each NADH is worth 3 ATP. So 2 times 3 is what? And then we have the transition reaction, 2 ATP. 2 times 3 is? 6. And then we have 6 NADH here. 6 times 3 is? 18. And then you've got your FADs. Those were each worth 2 ATPs, coming in a little bit lower. And you've got 2 times 2 is? 4 ATPs. So if we add up all these ATPs, it should be 38. So we've got 2 net. So help me out here. 2 plus 6 is what? 8. Plus 6 14. Plus 18. 32. Plus 2. 34. Plus 4. There's your 38 ATP. Heart and muscle cells are efficient like this, and for every glucose molecule oxidized, you get 38 ATP. Other cells of the body, for some reason, these glycolysis NADHs, because they're out here in the cytoplasm, all the rest of them have been made in here. Those glycolysis NADHs, they come in at the FADH level for some reason.